Good morning, ladies and gentlemen, distinguished guests, esteemed faculty members, respected students, and everyone present. Welcome to today's guest lecture on the theme Mitochondrial, Biology and Mitochondrial Medicine. To open this special occasion, let us begin with a moment of greeting and respect. Assalamualaikum warahmatullahi wabarakatuh. Peace be upon you, Om Swastiastu, Namo Buddhaya, warmest greeting to all. We are deeply honored to welcome the following distinguished individuals: Vice Dean Professor Dr. Hewan Aris Haryanto, our esteemed keynote speaker, Prof. Dr. Yidong Bai, PhD, representative from the collaboration and alumni, respected lecturers, researchers, and faculty members, and of course, all participants joining us today. Welcome to this lecture, proudly presenting a special seminar by Professor Dr. Yidong Bai, PhD. We are truly honored to have him here with us in here today, and we look forward to gaining valuable knowledge from his expertise in the field of mitochondrial biology and mitochondrial medicine. This guest lecture will be conducted in 3 sessions. The first session will be the main lecture where Professor Yidong Bai will share his insight, research findings, and perspective on mitochondrial science. The second session will feature an informative presentation about the graduate program at the University of Texas Health San Antonio. This is a great opportunity for those considering further studies abroad, especially in the biomedical science. The third and final session will be an open discussion and Q&A session. This will be your chance to engage directly with Professor Yidong Bai, ask questions, and explore the topic discussed in more depth. For those who have questions, we kindly ask you to save them until the last Q&A session, where we will provide time for a meaningful and interactive discussion. We hope you enjoy this seminar and that it sparked inspiration and curiosity in your academic and research journey. To formally commence today's event, it is my honor to invite Professor Dr. Hewan Aris Haryanto, MSc, Vice Dean for Research and Community Service, to deliver the opening remarks. Please join me in giving a warm round of applause as we welcome Professor Aris to the stage.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. Namo Buddhaya, salam kebajikan. Good morning everybody. Dear speakers, Professor Yidong Bai from Texas University, dear lecturer, dear student, all participants, ladies and gentlemen. On behalf of the Dean of Veterinary Medicine Faculty, Universitas Gadjah Mada, Yogyakarta, I would like to express my sincere gratitude and welcome you to the guest lecture on mitochondrial biology and mitochondrial medicine in Faculty of Veterinary Medicine, Universitas Gadjah Mada, Yogyakarta. In this guest lecture, I honorably welcome to our distinguished speaker, Professor Yidong Bai, PhD, from Departement of Cell System and Anatomy, University of Texas Health San Antonio, United States of America. The visit of Professor Yidong Bai in this faculty to represent a significant opportunity to foster international collaboration in research and education, especially in field of cell biology, anatomy, and mitochondrial medicine. Herewith, may I inform you about Professor Yidong Bai contribution to science, establishment of the first mitochondrial transformation system in mouse cell. The second is establishment of the cell model for mitochondria disease and development of therapeutics approach. The third is the role of mitochondria DNA heteroplasmy to tumorigenesis. The fourth is the role of the mitochondria haplogroup in human disease, including cancer. I hope this guest lecture today will be able to achieve the objective. First is to strengthen academic and research collaborative between Universitas Gadjah Mada and University of Texas Health San Antonio in USA. Second is to explore opportunity for joint research, faculty, and student exchange as well as collaborative academics program. The third is to engage with Universitas Gadjah Mada faculty member, researcher, and graduate student in discussion of the current scientific development. I wish you all the very productive in share in experience. I wish you all very enthusiastic attending this guest lecture, and you can discuss directly with the expert about the mitochondrial biology and mitochondrial medicine, which is interesting and exciting. May God bless us all with good health to make this guest lecture a successful and enjoyable one. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Professor Aris, for your warm and insightful remarks. Your dedication and continued support are truly appreciated, and your message has set a strong and inspiring foundation for today's seminar. Now, it is my distinct pleasure to introduce our esteemed speaker for today, Professor Dr. Yidong Bai, PhD. Dr. Yidong Bai, PhD is a distinguished lecturer in the Department of Cell System and Anatomy at the University of Texas Health Science Center San Antonio. He is a highly respected figure in the field of mitochondrial biology, with a deep and sustained interest in mitochondrial function across molecular, cellular, tissue, and animal levels. Professor Yidong Bai has made remarkable contributions in the area of mitochondrial biology, mitochondrial genetics, and also bioenergetics, with over 13 publications since 2023, a testament to his active and impactful research career. Maybe I would like to restate Professor Yidong Bai's major contribution, which include the establishment of the first mitochondrial transformation system in mouse cells, studies on the assembly of mitochondrial respiratory machinery, exploration of the role of mitochondrial DNA heteroplasmy in tumorigenesis, development of cell models for mitochondrial disease and therapeutic approaches, pioneering methodologies to create cell models with age and tissue-specific mtDNA for molecular and bioenergetic investigations. And lastly, insights into the role of mitochondrial haplogroups in human disease, including cancer. We are incredibly honored to have Professor Yidong Bai with us today, and we are certain his lecture will both enlightening and inspiring. Without further ado, please join me in warmly welcoming Prof. Dr. Yidong Bai to deliver his lecture. Let's give him a big round of applause.
Thank you so much. Good morning. First, I want to thank Dr. Aris Hayanto for the nice introduction and opening speech. And also, I want to thank the host for your excellent hosting, and thank Dr. Vista to facilitate my visit here. So let me start my lecture here. My title of this lecture is Mitochondrial Biology, Mitochondrial Genetics, and Mitochondrial Medicine. So let's start with, oops. Oh, okay. Okay. So let's start with this. This picture is really the EM picture of a mitochondria, which actually is in the cover page of Science magazine, which is a very prestigious journal, and in probably the turn of the century, and with an editorial titled "Mitochondria Made a Comeback." As we know that the first golden age for mitochondrial research is about around the 1960s. At that time, the merge or cross of cell biology and the biochemistry, both in the mitochondria. The biochemistry, we identify many metabolic reactions within the mitochondria, like TCA cycle, biosynthesis, or pyrimidine, amino acid, and also cell biology, just because we developed very sophisticated technology Number one, we know how to fractionating different compartment of cellular organelles or organelles like mitochondria. We can visualize the mitochondria. So, and also we have a high-speed centrifugation and plus this, the EM. So we can visualize what this mitochondria look like. So that's the first golden age. Second one is about now. It's also the merge of several disciplines, including, and we study the population genetics, right? Really what we do is we sequence the mitochondrial DNA sequence from the samples, people all over the world. Then we compare those sequences. So that if you have a mathematical mind, you can build up a tree. So that means if from the same area, however, the sequence quite diverse, that probably means that they are very ancient. They have diverse to begin with. So with that, so people come up with this idea of African Eve. So they believe, so all current human beings are all originally coming out of Africa. Then they migrate to Eurasia and further all over the world, right? So then also a lot of human disease has been associated with mitochondrial dysfunction. Early time, we call this mitochondrial disease because it's number one, they notice some patients has some intolerance of the exercise. So they cannot catch up if they work too hard. And number two is that under the microscope, they see the abnormal mitochondria, enlarged abnormal ones. So they think those people with mitochondrial disease. However, with not expanding our knowledge about mitochondria, we know many common disease like metabolic disease, diabetes, like neurodegenerative disease, Alzheimer's disease, or Parkinson's disease, or even cancer. They all have some sort of mitochondrial dysfunction. So then they come together, and also we study the molecular biology, and we know the initiation of cell death or apoptosis. They also start within the mitochondria. So again, everything mitochondrial medicine, human population genetics, and molecular genetics, they come together within the mitochondria. They create this second, we hope, still going on, the golden age of mitochondrial research. Oops, sorry. So let's start very beginning to show you, convince you how important mitochondria are. So starting from energy metabolism, as we know, the food you eat in mostly in the form of the sugar, or they digest in your GI tract, they end up with glucose, right? And the fat, they digest, produce fatty acid. Then they utilize different, I must touch something. Okay, thank you. Yeah, thank you. Very helpful, thank you. So we have the different receptors mediate the transport from the fuel from the circulating system of blood into the cells. And here, glucose into the cell, then goes through glycolysis, then generate the end product of pyruvate. Then they have cross-membrane transport into the mitochondria. Then they going through further oxidation to generate acetyl-CoA. Similarly, the fat getting to fatty acid facilitated by a particular receptor into the cell in the form of fatty acid. Then the fatty acid, again, with a particular receptor, mediate the transport into the mitochondria, then goes through a fatty acid oxidation. That's also happened within the mitochondria. Then they generate similarly acetyl-CoA. Then the acetyl A will enter the CTA cycle, and then this CTA cycle will generate NAD, which is the major fuel or the electron carrier enter the electron transport chain. Then the majority of ATP, we know ATP is the energy currency, right? Then will produced by oxidative phosphorylation, which is the oxygen transfer along the respiratory chain in the inner membrane of mitochondria, which were coupled with ATP synthesis, which is phosphorylation, by the membrane potential, which is established by the energy convert. When the high energy electron transfer downhill, the energy will be utilized to pump the proton across the inner membrane of the mitochondria to establish this membrane potential. Membrane potential, as we know, is energy storage form, which further will be utilized by complex 5, which is the ATP synthesis, to produce ATP. Let's show here, the oxidation process is really the electron transfer, right? Starting from NADH to NAD, which is happen at complex 1. Then they further electron donate to ubiquinone, and then further donate to complex 3, and mediate by the by cytochrome C to complex 4. Then at the complex 4, the relatively lower, which carry the lower energy electron, will meet with the oxygen to produce water. At complex 1, 3, and 4, there will be a proton transport, as we just mentioned, across inner membrane of mitochondria, which will establish a membrane potential or the proton gradient. The proton gradient then will be utilized by complex 5 for the ATP synthesis. So this is really one of the most beautiful biology model, right? As if I ask you, the student, what do you think is the most important biology model in past century? If anybody can give an answer. So which actually the model is important is they will solve an important observation, right? So if I said it's the genetics, if you can remind you something. So the genetics is really people observation that from generation to generation, like father, like son, like mother, like daughter, right? There must be some genetic material across the generation. So what could be the genetic material, right? So at the very beginning, there is a controversy. Some people think it could be protein, right? And also a minority at that time think it's the DNA, right? Then it is until Watson and Crick, they come up with this model called double helix. You know, there's 2 strands of DNA string. They complement to each other, right? So this double helix model can clearly explain how this genetic happened, because the 2 strands will separate, then complemented the synthesis to generate 2 daughter strands. So that's so they have then passed to the next generation. So that's the base of the genetics, right? But this is the model. Double helix is really intuitive. That means it's quite straightforward. You can relatively easy to appreciate. However, this oxidative phosphorylation to explain the ATP synthesis and the respiration, which is actually the electron transfer to oxygen is relatively not that intuitive. There is some twist. So that's probably the reason this series, thank you so much. It take time for the series get accepted. And with that, Dr. Peter Michel, who get the Nobel Prize in 1978, it take about 20 years. It's not like a double helix. It probably take only 5 years get recognized and get Nobel Prize. So the statement for his contribution to understanding the biology energy transfer through the formation of this oxidative phosphorylation theory. So I really want you to appreciate more of this theory so we can further look into this, or the one name for it is chemosmotic coupling hypothesis, right? So it means a proton gradient, electron chemical gradient, right? It is also established by electron transport is driving, this proton gradient is driving oxidative phosphorylation. The electron transport complex and ATP synthesis are in the physical distinct region of the membrane, which they are responsible for two distinct process: electron transfer, which is oxidation, because finally deliver the electron to oxygen, ATP synthesis, phosphorylation, responsible by the complex 5. They are in separated regions. And also, it means imply a vectorial metabolism. That means this reaction, this imply an enzyme embedded in a membrane in a unique orientation might characterize a reaction by accept substrate on one side, which is matrix, right? You have the inner membrane, the really one taking substrate on one side, then they release, however, the product on the other to the the intermembrane space. You have the inner membrane, you have matrix, and the intermembrane spaces. So then they generate the membrane potential. And also they imply, so these are relatively old revolutionary idea. So microscopic, the osmotic process convert to macroscopic osmotic process. That means you have the pumping at the different places of the membrane. Then they add together to generate the overall membrane potential, which then be utilized by another enzyme system, which is ATP synthesis. So during this time, the free energy change associated with such reaction might be used to build up gradient with the enzyme both at complex 1, 3, and 4 as a pump, right? A concentration gradient of solute across a membrane could be considered as storage of chemical free energy, which is also very novel at that time. So let's look at this, how that happened. Again, this is complex 1, which is the L structure, which one arm protrudes into the matrix, another arm sitting in the inner membrane. As you see here, they have different, the domain of the enzyme doing different things. For example, the membrane arm responsible for the proton pumping to establish the membrane potential, and also the matrix arm doing the catalyze the reaction, redox reaction, to change the NADH, to accept electron from NADH, then release NAD. And also here, so they donate electron to ubiquinone, and ubiquinone to donate electron to complex 3, and complex 3 donate electron to another electron transfer vehicle, which is a cytochrome C. Cytochrome C then donate the electron to complex 4, and there they meet with oxygen. Right? So that's what happened. So probably some of you may note there is one very interesting molecule called cytochrome C, which is certainly an important molecule for electron transfer. However, probably some of you are also aware it is an important molecule to control cell death. Right? Once this molecule released from the mitochondrial, they go into the cytosol, then can initiate the cell death process. So this is the one of the. So also, I really want to remind all of your students that biology concepts are really all evolving, right? So for the undergraduate, probably your task is memorize, right, all different theories. However, when some of you keep going on, go to graduate school, for you then will try to identify some of those concepts probably worth revisiting. Probably you suspect with the new technology or new system available, those knowledge could be replaced by new one. For example, here is one about the organization of this respiratory chain, right? So as we discussed, pretty much electron transfer going through complex 1, CoQ3, cytochrome C4, and however, there is some problem if really they work individually and independently. So one thing is really the efficiency, right? So if you can imagine sometimes the electron, instead of delivered to CoQ, if this physically separate, you have a good chance for those high energy electrons will leave the electron transport chain prematurely. Then instead, they will go all the way to complex 4, they will leave, then they will meet with oxygen, then they will have you anticipate the reaction which will generate free radical, right? As you probably know, free radical has been proposed as the cause for some pathological consequence, and also the efficiency, right? So probably not all the energy carried by the high energy enter the chain will be utilized to pumping the proton to generate the membrane potential. So alternatively, early on, some people proposed the idea called all those membrane, the complex, probably they come together to form a higher organization called supercomplex or respiratory zone. Right? So then that will make electron transfer much efficient. So I will probably discuss more of this concept tomorrow in my research lecture. So again, this not has been able to prove until we have some developed or better method, which including we find a better detergent which to separate those proteins, enrich isolate protein, which is detergent, the digitoning. And also we have a better way to separate those molecules, which we use a blue native gel, which is electrophoresis system. So as you can show here, really with this detergent, with digitoning and blue native gel, we really can visualize some of the molecule membrane proteins complex, which have a relatively high molecular weight, which is corresponding to those different complex come together. Then we can validate it by 2D gel, further separate the individual subunit of the complex by secondary SDS gel, Then we can do Western to determine really some of the high molecular weight complex contain the subunit coming of different complex. And also we have other technology which including the electron microscopy. We can actually visualize those compound. Its form is look like multiple complex come together. And also with the aid of supercomputing capacity, we can resolve the structure as shown here. See here, this structure is L structure with complex 1, and the red one like complex 3 together, 1 and 3. And here is 1, 3, and 4. So we identify those higher molecular structure with multiple, more than one respiratory complex combined together. So this is just the cartoon show that, so we have complex 1, CoQ, complex 3, and 4 together form a supercomplex to facilitate the electron transfer, or we can call it respiratory zone, because this one single complex can perform the respiration from the beginning to the end. So then I will switch gear a little bit to discuss mitochondrial genetics. Mitochondria actually is the only organelle in animal cells which contain their own genome. They have their own DNA. However, this in mammalian cell, the mitochondrial DNA organization are very, very conserved. They have almost, this is a representative human mitochondrial DNA, about the 16.5 kb. It's relatively small, and they encode 13 peptides. All those 13 peptides are the subunit of the respiratory chain complex. 7 for complex 1, 1 for complex 3, 3 for complex 4, and 2 for complex 5, which is ATP synthesis. And interestingly, they also encode 2 RNA and 22 tRNA. So as we know, RNA, tRNA are the translation machinery. So that means mitochondria, they have their own translation machinery within the mitochondria, which actually is quite different from the cytosolic translation machinery. That's probably, evolutionarily speaking, I don't know, you had the idea, is they propose the mitochondria is really originally coming from bacteria. That's one evidence to support it, is because this machinery mitochondria is more like a prokaryotic, like an E. coli. They're very different from the cytosolic one, which is eukaryotic. So which is good, actually. We can utilize this difference, then use its difference, the sensitivity to some of the antibiotics. Then we can use different inhibitor to, when we're doing the research, we want to visualize the translation for cytosolic only and the translation for mitochondrial encoded protein only. So we can distinguish to see each contribution in different scenario. So this, I read just the list some of the features of the mitochondrial genetics, unique features make it fascinating. And mitochondrial DNA, so although we always think of talking about genetics, you are 50% from your father, 50% from your mother. However, strictly speaking, genetic-wise, your mother contributes more than your father, just coming from mitochondrial DNA because mitochondrial DNA are predominantly maternal transmission. That means your mitochondrial DNA from your mother only. Your father has no contribution whatsoever in this case. And also, compare with nuclear DNA, the nuclear DNA, we know that when you study the cell cycle, you have two alleles, right? You have to make sure they replicate once during cell cycle, replicate once, but only once. However, mitochondrial DNA are really a population. They all sometimes have hundreds or even thousands of copies. So they contain this. So that, interestingly, they're coming up with the term called heteroplasmy. That's oftentimes associated with the pathology, the condition associated with mitochondrial deletion, the mutation. That means sometimes, in some case, or oftentimes, within a cell, the mitochondrial DNA are not all identical. So when they are different, we call those kind of status called heteroplasmy. When everything identical, we call homoplasmy. So as you can think, when the mitochondrial mutation can accumulate, right, cross certain percentage, we call threshold. When at this, we identify this status like they cannot, because you have too many mitochondrial DNA mutation, which cannot, it's end up you have too many dysfunctional protein, end up with, you don't have enough functional mitochondrial machinery to support the normal physiology, then you have this dysfunction or pathology in particular cells. So that's also this mitochondrial DNA replication, inheritance, stochastic, and different the cell and tissue. So that's why in some case, if you have the mitochondrial DNA mutation, however, oftentimes you have tissue-specific presentation. So this tissue-specific presentation, the reason for it could be have twofold. Number one is probably in different tissues, they have different demand or sensitivity. So number two is coming from here is the heteroplasmy. Just because of the replication, segregation, then end up in certain tissue, certain cell, they have more mitochondrial mutations. Then they will show up some pathology. So again, through this, and also there is another example show you that concept, the theory could always evolving. In this case, is mitochondrial DNA metabolism or expression. So people think, probably 10, 20 years ago, people think mitochondrial DNA are changing faster, or you have easy to accumulate mutation because mitochondrial DNA compared with nuclear DNA. With nuclear DNA, you probably know that they have a histone structure. They package the DNA into histone to chromosome, right? However, they said they always describe mitochondria as naked, floating, and now we know that it's not true. Mitochondrial DNA actually is also packed into nuclear structure, that they have some protein, particular protein, for example, TFAM, transcription factor, translational regulators, they come together, then packed mitochondrial DNA into an organoid structure. So that, in one way, they make it well protected in some sense, and also all those process to mitochondrial DNA is highly regulated. So then we go down to the pathway. you have mitochondrial DNA, then you have the replication, and for gene expression, you have the transcription, right? Transcription means you have the DNA, they will generate RNA. RNA is the template for protein synthesis. So here is to show you how the transcription happens. So again, here, more like prokaryotic system, you only have two promoter here, heavy strand and light strand promoter. Then they generate a large transcript. They go all the way through the genome. Then after that, when you have this RNA processing to process the big transcript to generate multiple transcript, which will serve as template for translation. So then here's the translation. Again, as we said, the inner membrane, right? Inner membrane where the oxidative phosphorylation happened there. Again, here is another important process called translation, also happened on the inner membrane, where ribosomal biogenesis, then you have the mRNA will recruit into the translation machinery, which is ribosomal. Then they further recruit tRNA, which carry the amino acid for protein synthesis. Here, just give you an overview. We have the mitochondrial oxidative phosphorylation, and we have multiple complex for this process. And both mitochondrial genome and the nuclear genome contribute to the oxidative phosphorylation structure. As we discussed, particularly for complex 1, you have 7 from mitochondrial DNA encoded subunit, the other 38 or 36 from the nuclear DNA encoded. For complex 2, all 4 encoded by nuclear DNA, and 3, 10, and 1, 4, 10, and 3, 5, 14, and 2. So as you can see here, as we say, mitochondrial DNA is important because they encode essential subunit for oxidative phosphorylation. However, having said that, majority of those subunits, the structural component, still encoded by nuclear genome. So again, here give you an overview. So we have the mitochondrial DNA, right? Then they encode essential subunits of the respiratory chain. Then you have the other subunits encoded by nuclear genome. They come to mitochondria through transport system. Then there's 2 type of subunits assemble into the respiratory complex or respiratory chain at the inner membrane of mitochondrial formed oxidative phosphorylation system, which responsible to generate ATP or energy metabolism. This is important process to keep our different organ functionally at, for example, it's pancreatic, kidney, liver, eye, heart. So all our organs, probably most for those energy demand is high, like brain, heart. So that's probably the reason you probably have more chance to have mitochondrial dysfunction associated disease with like brain, heart, or muscle. So this here just show you it's because mitochondrial function is so important. Any disturbance, any dysfunction mitochondrial DNA can cause disease almost all over your body. So here is one example of mitochondrial disease, which is called the Leber's hereditary optical neuropathy or LONE, which actually is the first disease has been identified was associated with mitochondrial DNA mutation, which cause blindness. You can see here is imaging to show. So here's the summary of the feature, which is subacute to acute loss of vision, blindness. Mostly happen to men. Age onset is about when you are very young, and clinical feature of optical neuropathy, and you see this degenerations, right? Okay, so this is also summary of. Okay, so this degeneration, sudden onset. You have the primary mutation for this Leber's hereditary optical neuropathy is really the 3 mutation in mitochondrial DNA encode subunit for complex 1. And sorry. This is the vision. So those patients, peripheral vision is okay, but they lost the central vision. So that means this mutation, particularly also you carry this mutation all over your body. However, most affected the area is the retinal ganglia cell, so which is responsible for the central vision. So so here is again the mitochondrial DNA caused disease. So mostly the maternal inheritance. So how if you future practice medicine or even with a vet, if you see a patient, they have a maternal inheritance. That means you look at check the family tree, you see always the maternal relatively show this particular phenotype. You probably can, or it's the particular phenotype of the disease is something to do with energy. You probably suspect it could be caused by mitochondrial disease, the mitochondrial DNA mutation. The easy solution is just sequence the whole genome, right? So another one to help you to see if there's any possible mitochondrial disease is the histology, right? You have particular staining method. If you see this muscle called red fever, which is show the abdominal mitochondrial, is for this mitochondrial myopathy. It's also a hallmark for the mitochondrial disease. So again, I will show we have a tremendous progress, accumulation of knowledge. It's when back in 1988, that's when we first report about the mitochondrial DNA mutation has been first associated with human disease. At that time, one is the Leber's mutation, which caused blindness, which is happened to the mutation in complex 1 subunit. And the other mutation identified at that time is really a large chunk of deletion or common deletion, which has been associated with neurological disease. So although mitochondrial disease or mitochondrial DNA associated disease has been identified many, many years since now to now, still we don't have a full understanding the mechanism study. So there is no, for many of those the disease, we don't have the good treatment. So that's the show summary why it's very complicated. So it's oftentimes the same mitochondrial DNA mutation may not produce identical disease. So that means they probably, on top of mitochondrial DNA mutation, they have other players or modifiers, right? They participate on top of mitochondrial DNA deletion. And also, interesting, different mutations in mitochondria and nucleus can lead to the same disease. Probably that's not that surprising because they probably participate in the same process. Right? So, although it could be, as we summarize, you have the mutation, probably all of the genome can cause trouble. However, we do have some hotspot. That means oftentimes some of the mutations in mitochondrial DNA encoded tRNA are more likely associated with different diseases. Here is one example we call MILAS mutations, which is happen at mitochondrial DNA encoded tRNA losing UUR. So here is to show you only up to 2010, more than 200 mutations has been identified. So, and also this here is our understanding. Mitochondrial dysfunction in preclinical disease, although the first, when the first mitochondrial disease has been coined, come up with the name, this, at that, back in 1962, at that time, everybody believed it is a rare metabolic and neurological disease. However, as I said, also now many common deletion, the condition including neurodegenerative disease, metabolic disease, and even normal aging or mental disorder has been associated with mitochondrial dysfunction. And again, also people think those mutations many years ago are very rare. However, as that population study recently carried in Europe, and that we found that actually there are many silent mutations. That means many people, you carry this mutation, but not necessarily just waiting there until it's chance to cause trouble, right? So it's not that because of the disease, at that time believed it's only 1 in 500,000. However, they found more than in 1 in 5,000 people, they do carry pathogenic mutations. So on top of that, not only those pathogenic mutations, that the mutation has to be established, those mutations can encode, disrupt the process, right? But even those mitochondrial genome type, as we discussed, when people migrate out of Africa and other places, they accumulate some variants. During those human migration, those variants fixed in certain population. And recently, we call those genotype, right? Particular defined different genotype or haplogroup. And the recent people has been also associated those genotype as a risk factor or protective factor to different people. So this is to show this human migration we already discussed. And during this human migration, some of change are fixed. So here is the example. For example, there is one haplogroup or genotype called F4, which show that more likely have the obesity, right? It was a good p-value. And the D5, on the other hand, also with this, with particular, again, it's metabolic syndromes. So another concept, how this mitochondrial mutation or dysfunction can regulate this process. So here is the mechanistically, there's a possibility. So if there's something wrong with the mitochondrial, of course, there's a crosstalk. Right? As we said, the majority of the mitochondrial structural protein encoded by nuclear genomes, and all the regulatory protein mitochondria encoded by nuclear genomes. Right? There's one regulation from nuclear to mitochondrial function. However, there's another way, the back from mitochondria. If there's something wrong, mitochondrial dysfunction, they can really reprogram the extranuclear system through this we call retrograde signaling. The mediate could be calcium, ATP, ROS, free radical, acetyl-CoA, or even redox, NAD and ADH. So at the last part of the lecture seminars, I just want to introduce you some approach we study mitochondrial function. So in the future, during your study, if you observe something you suspect could be involved mitochondrial function, or it could be something caused by mitochondrial DNA mutation, you probably can try those different methods. One is called generate cybrids, which is a method to separate mitochondrial DNA and the nuclear DNA. And also, as we discussed, the machinery of translation of mitochondrial and cytosolic are quite different. You can use this to distinguish, to see probably which part could be wrong. And the blue native gel we discussed to analyze the respiratory chain. And also, because of the mitochondrial oxidative phosphorylation, quickly you want to check if the cell is there some mitochondrial dysfunction, you just measure the oxygen the consumption. Have a quick assay. Very quickly, here is how we separate this the genome. So what we do is we just have we try to have a method to move the mitochondrial DNA around. The rationale is we want to separate this two genome. Then, if we want to follow it to see the particular dysfunction, will go with the mitochondrial DNA or go with the nuclear genome. If they go with the mitochondrial DNA, more likely they probably carry the mitochondrial DNA mutation, right? So what we do is really in one donor, we call mitochondrial donor, what we do is we disrupt the cellular skeleton. So then we do centrifugation to enucleate. That means the nucleus cannot be supported by the skeletal system. Then you will end up with a cytoplasm, right? With cytoplasm, no nuclear, only mitochondria. In another, we call the mitochondrial recipient. That means we want to get rid of the depleted mitochondrial DNA. What we do is we have particular chemical, ethynyl bromide, for example. They relatively enriched in mitochondria because of the charge, right? Then they interact with DNA to block the DNA replication. Then over the time, you have a rho-zero cell. That means you deplete mitochondrial DNA. Then you do the cell fusion. Then we obtain a trans-mitochondrial line. So here is the example. We utilize the inhibitor, which can inhibit the cytosolic translation. Then we do protein synthesis labeling. The only mitochondrial DNA encoded 13 peptide will be labeled. So then we run the gel to look at the see if any particular band will change. Then if they disappeared or decreased, then you have a chance that there could be a mutation there. So you have a blue native gel, 2D gel. So here is the example. You do the oxygen consumption. You probably heard the seahorse. There is a very sophisticated machine can measure the change of oxygen concentration or measure oxygen consumption. And it can go further. You coupled with the permeabilized cell. The reason you do permeabilized cell, then you want to get rid of the endogenous substrate. Then you provide the cell with specific substrate. Then you can measure the oxygen consumption. Then this oxygen consumption is based on the particular substrate activity. For example, you use molyglutamate, then you measure oxygen consumption, you measure complex 1. And similarly, you have succinate, G3P, coupled with the rotenone, which is complex 1 inhibitor, you measure complex 3, and similarly, you can also complex 4. So that's what you are doing the measure here is you in a chamber, you measure the change of the oxygen concentration. And when you put the cells, you have a stopper, right? You see the oxygen concentration decrease, which because it's consumed by the cells, right? Then you put inhibitors, you just block the oxygen consumption. Then you put, in this case, you put secondary substrate. Then you resume the oxygen concentration. So this here is just give you the summary. I just borrowed Doug Wallace. So originally, we understand, we appreciate the contribution of mitochondria is because their importance in generating ATP. Then, with our understanding of mitochondrial function increased, we know mitochondria also played other important role, cellular role, on top of energy metabolism, and also they can regulate really the cell death, right? And also by generating, we didn't discuss a little bit, is that you can also generate reaction oxygen species, which is an important molecule, can initiate many reactions, and also mitochondria is an important regulate for calcium signal. As we know, calcium is secondary messenger, right? So altogether, we think mitochondria is really, in some sense, play essential role to control the cell death and the cell survival. Oops. So I just probably. Oops. Okay. Probably at the very last, I want to show some picture of San Antonio. If I go to the very last. Okay, so here is we have this riverwalk. Is the San Antonio is the tourist destination. We have a European style of riverwalk. We have by the bank. This is the Christmas time, and this is the regular time. We have those restaurants by the bank, and also we have also some historic sites. This is a Spanish mission. And also here is what we call Alamo. I don't know whether any of you watched the movie Alamo. We're really, Texas historically is part of Mexico. And until there's a war happened in this church, then Texas became independent. Then, of course, we joined U.S. as the one of the state. But here is the historic site, Alamo. And here's the overview of our medical center. Okay, thank you.
Okay, firstly, I would like to say thank you so much to Professor Dr. Yidong Bai for the first session of the presentation. So as we can see that the mitochondrial has a very special and very important in our life, very small aspect, but it leads up to a very important function in our human body. Okay. Now we will move to the second session of today's seminar, which include information about the graduate program at the University of Texas Health Science Center, San Antonio. This is a wonderful opportunity for those of you considering further studies, especially in the biomedical science. To learn more about the academical environment, research opportunities, and pathways available at one of the leading institutions in the field. Please stay engaged and feel free to take note of any questions you may have. They can be addressed during the upcoming discussion session. Okay. Thank you so much, Professor.
Okay. Thank you. So then I'll keep going on to introduce the graduate program available in our institute. So I will start with UT. We are part of University of Texas system. So University of Texas, they have 14 institutes, including here is probably we have a six medical center and 8 academic institute. As you probably heard, UT Austin is really the flagship institute. It's in the capital of Texas. And they also include the UT Arlington, UT Dallas, UT El Paso, UT, this is the new one, PB, and the Rio Grande Valley, and the San Antonio in our city, and Tyler. And also we have 6 medical institute, health institute, including UT Southwestern, which is in Dallas, UTMB, Galveston, which is the port city close to Houston. And we also have the Houston, San Antonio, and the Anderson, which is the important cancer center, and the UT Health. This is the new one in Tyler. So San Antonio, again, I already touched a little bit about the San Antonio. We have another name. It's called the Alamo City, because we have the Alamo, which is a very historical site, and it's important in not only Texas, but also in U.S. history. Really, San Antonio, probably you didn't heard this, we are actually ranked number 7 in the nation in terms of population. And one of the fastest growing city in the country. We have population keep growing. We have some Marathi majority. That means we have a faster growing Hispanic population in our city. And we have a rich and a vibrant culture. So you can have different types of meet together. Relatively, we have a lowest cost of living, right? There is a comparison. So for those, if we have 30,000, for example, in here, it's comparable to almost 50K in other places. So it's a very livable places. So our institute. So here is, we have academic health center with 6 schools. We have all 6 schools together with, of course, the graduate school, where PhD, master's degree. There is, we call graduate school of biomedical science. And also we have medical school, which is mostly for MD student, dental school, nursing school, health professional, and public health. And so we are best known, quite famous for research in aging, since we have Gadjah Institute for Aging, and also for cancer study. We are one of the comprehensive cancer center, which I think nationally only 50 or 60. And also we have quite famous for cardiovascular study, neuroscience, diabetes, infectious disease, military health, because another name for San Antonio is Military City, USA, and the regenerate medicine. And here we are one of the Hispanic Serving Institute. So UT Health, we certainly, as we discussed, we have a logo, motto, we make life better. So we certainly have a graduate school among, as we just discussed, other schools. And within the graduate school, we have called IBMS, interdiscipline biomedical science. So we have different track, different discipline. So we have physiology, pharmacology, we have biology of aging, cancer biology, cell biology, genetics, molecular medicine, neuroscience, biochemical mechanism in medicine. So among this 3 biology of aging, cancer biology, and they are affiliated with our department. So some of the research highlights, we have totally this amount of funding annually, so more than 400 million total sponsor program awards, research. So we have this is relatively new, Green Biggs Institute for Alzheimer's Neurodegeneracy. And also we are the leading institute to study for PTSD, which is the post-trauma syndrome, the disorder. So it's for battlefield, some of the soldiers coming back from the battlefield, they always have this kind of conditions. We also have a Bishop Institute for Longevity and Aging Study. We are one of the NCI designated cancer center. So we also have, this is actually supported by tobacco money. So there's a big settlement about 20 years ago. We use this money to study, to establish Children's Cancer Center. And we also have these military, America and Military City, USA. So we have a good association with Military Institute. So here is the sum of the reason we're standing out. In 2024, Nature Index ranked UT Health San Antonio number 6 on the list of fastest rising institute in United States for research output. We are only one of 11 in US with NIH-funded NADH and DDS and PhD. We have 2 double degree funding. And we have multiple basic science department ranking 20 and 30 in terms of funding, so we are one of them. I think both biochemistry, cell biology, our department, I think we are ranked about 23rd or something, and the pharmacology at the 3 and 4, they are ranked among 20 and 30. And we have over 250 faculty members with 400 million annual sponsor program in research activity. We have 10 pre-doctoral research training grants to support IBMS students, including NIH sponsored for the IMSD, which is quite prestigious. Our IBMS program offers exceptional, flexible to students, offering the opportunity to choose from many laboratories, different areas of science for PhD study and open for collaboration. So IBS, again, data. So approximately, annually we got 400 to 700 applications, and average GPA is domestic one 3.5, but we have a minimal. If you 3.0 is the minimal, then you can apply, and we usually invite 80 to 90 for visitors. For international, we usually contact phone interviews. For each year, we accept around 40 students, and the student body are quite diverse. And media time for the PhD degree finished, the average year is 5.3, and relatively shorter than the national median, which is 4.4, 6.4. Okay, so the support. So all students receive, IBMS students receive 35K for annual stipend, and also the tuition fees and insurance are also covered if you accept it. Usually you would expect the progression, the journey. The first year you have the core course, and also you do rep rotation until you identify a mentor. And then in the second semester, you're doing the advanced course. So then into the second year, you're still doing some coursework, but then you mostly switch to research. And the second semester, you have to do the qualifying examination. Once you successfully pass the qualify, you enter the candidacy. Then you propose, in the third years, you propose and form a thesis committee. And then after that, mostly you focus on research. Right? You finish the coursework. But that time you also participate the seminar, journal club, and also present, expect to present at scientific conference. Then by the end, hopefully by the end of 5th year, you will defend your thesis. So here is, then I give you an example of biology, the aging discipline. So we have a building for Bashop Institute, which is mostly the aging faculty host there. So we do have basics biology, and we also focus on sarcopenia, dementia, and we have some interesting the models, which including probably tomorrow I'm going to make it more rats, which is a small rodent, but they can live up to 30, 40 years old. And also we have some clinical trials. And also faculty there, we also have stem cell research, DNA repair, and the metabolism of aging, senescence, inflammation, Alzheimer's and Parkinson's disease, interventions, care restriction, pharmacological therapies, behavior modification, and we have a multiple clinical trial involved. So here is the faculty member of our institute. I think hiding somewhere. So here is the okay. So this is the physically. We have also some course within the Aging Institute. So that's we are only in the nation have 3 NIA, National Institute Aging Clinical Research Center. And here is the intervention system. That's the one center is called by Nathan Shock Center, which is the basic aging research. Nationally, only 6 or 8. And also we have an intervention test center, which is for anyone who would come up with a magical molecule or compound we think it will extend our lifespan, we are going to test it for you. Then, of course, after screen, peer review. And we are among 4 other centers will test those, the compound. And also we have doing some human studies. So we do have weekly seminars. And also we have annual the conference in this ranch, Maya Ranch, which is close to San Antonio, to discuss the aging research. So there's different working. So here is this is the PhD. So we also have a master's. So this is managed by our department. So we have usually we can decide which student to take, and so we have 2 tracks. One is mechanism of health and disease track. The other is anatomy science track. So for this one, they always 2 years. You are doing a thesis. So this is the outcome for our master program. So about 20% of our graduate from the master program will go on into PhD, and 50 about those graduates enter medical or PA dental professional school. About then, 17 of those graduates were doing lab research as the technician or research assistant. And so 7% will end up with teaching positions. Okay, so I think that's all I want to introduce. I'm happy to answer any question if you have. Thank you.
Okay, thank you so much, Professor Yidong Bai. For the second session, we truly appreciate in the insight you shared about academic opportunities and pathways for aspiring researchers and also graduate students. Now we are moving into the final session of today's event, the discussion and the Q&A session. This is your opportunity to engage directly with Professor Yidong Bai, ask questions, and dive deeper into the topics that have sparked your interest throughout the seminar, whether it's about his research, the graduate program, or broader topics in mitochondrial biology. We welcome your curiosity and participation. If you have any question, please feel free to raise your hands, or if prefer, you may write it down. Dan apabila ada yang ingin memberikan pertanyaan dalam bahasa Indonesia, juga sangat diperkenankan ya, teman-teman. Oke. Apakah ada pertanyaan?
Hello, good afternoon or good noon. Yeah, good morning, I think. So my name is Nuriana Vendita. I'm practicing small animal practitioner, and actually I just want to ask something related to the mitochondrial DNA. So actually the mitochondrial DNA itself, it's inherited from the mother, yeah? It's like 100% inherited from the mother. And if, let's say, the mother is aware, if she actually carries this faulty or, the defect of this mitochondrial DNA. So I think that's also possible if she doesn't want to pass this to her offspring. So then she can actually, from your explanation before, actually doing the donor, mitochondrial DNA donor. So then actually she can get, like the healthy, like the oocytes with healthy mitochondrial DNA, and then put her nuclear DNA there, and also the sperm from the father. So in the end, it's gonna create, like the embryo with 3 different DNAs, right? From the mother, like the nuclear DNA from the mother, nuclear DNA from the father, and also the mitochondrial DNA from the healthy donor. That's the case of embryo. And my question is, if the disease is already established, how's this process will help? Like this mitochondrial DNA donor will help with the disease that is already established. Whereas, like before, it's like we are actually setting an embryo that will not have, complication in the future. So my question is, if the disease is already established, how is it with this mitochondrial DNA donor? Thank you very much for your answer. Thank you. Good morning. Thank you, Professor.
Thank you for the excellent questions. So one thing is certainly involved with genetic counseling, right? When a new married couple coming to a genetic counselor, they want to say, we want to have a baby. However, then the mother carry a particular mitochondrial DNA mutation, what we should do, right? So at that time, so one thing is, as you suggest, there is a sort of like a three-parental strategy, which has been tested, I think in US or in China, even Britain makes some news. They actually have the mitochondrial DNA, you deplete the egg, the mitochondrial DNA from the mother's eggs. Then you fuse the white and the functional mitochondria from a donor. So then you have this sperm coming in. Then you have this offspring, which actually coming from 3 parents, right? So that can be done. However, if the baby already born, which certainly, as you said, carry this mitochondrial DNA mutations. So there is a one way called mitochondrial transport. They actually can inject here healthy mitochondria into the disease area, be it brain or muscle. So then the idea is through this endocytosis, they can take this healthy mitochondria, hopefully those who are taken over. So again, as I said, although mitochondrial DNA is important. However, it's oftentimes is not really 100% prevalent. So you still need something else to make the disease happen. So there is a possibility, even you have this mitochondrial mutation, not necessarily will develop disease. That's the case, for example, for those typical mitochondrial disease with the same siblings, only several of them will develop this disease. Others with the mutation, they are okay. And also, I said this is interesting because it's originally so those typical mitochondrial, classical mitochondrial disease. This should be 1 in 500,000. However, if we look at the DNA level, those really carry those pathogenic mutation. It's much more, even 1 in 5,000. So that means probably majority of case, you have the DNA. That means you're doomed. So probably those are the silent one. But it could be still transferable. It's sort of like a silent bomb there. It could, you really want to do something. Probably that's a good idea. Thank you. Thank you.
Terima kasih kepada FKH atas acara yang bagus ini. Sangat menarik. Nah, let me introduce myself. My name is Meta. I have several questions. Could I? First, how important are mitochondria function comparing to all immune system in human body? That's the first. And the second, what is the food for mitochondria at normal condition? And the third, how to boost their mitochondria, I mean stimulant, in a safety or complex, in a safety way when there is a disease or complex diseases. And the next, could you please sharing, Professor, about abnormal mitochondria and give the more knowledge, I hope in detail, about the explanation of abnormal mitochondria. And the next, sorry, I have many because at this moment I'm working to handle one product that's herb, and it's work also for the immune system for animals. So I really need your. And the next is based on your expertise and observation. Is there any condition permanent damage of mitochondria, and what is the causative agent? And the next, normal aging of mitochondrial dysfunction since. Thank you, Professor, and thank you for coming to Indonesia.
Oh, thank you. I hope I can remember all your questions, but let's do one at a time. But if I forgot anything, please remind me. So for this, but it seems like you're from your personal interest mostly on immunology, immune response, right? So that's how mitochondria involved is. So as one thing, of course, there's a particular immune response, like a step, like a mouse protein, which are actually coming to mitochondria. And in particular, those they call the immune response. Some of those immune response involve viral infections, involve some of the viral encoded proteins getting to mitochondria that initiate something there. So on top of that, I know one certainly the inflammation. So it's also something to do with the immune response, right? So now we believe mitochondria play a key role for this inflammation. So there's several proteins at the inner membrane has been identified, which is is mediate the inflammation. As we know, inflammation is also probably play an important role in aging process. So that's what people mostly focus on is this acute immunoresponse, which involves outmembering some of protein there, can mediate immunoresponse. It's mostly mediate. So one thing is some sort of the cellular process involve mitochondrial. I think is nature is smart because mitochondria, they form a network. For those immunoresponse, this is the ideal system, right? You have the signaling coming in, being it was this infectious or other exogenous things coming in, invading, then mitochondrial is getting respond, then they have a network, so they can sort of organize this response. So that's the one way I understand. And so what's the second question? So, about aging or DNA abnormal mitochondria?
What's the role of mitochondria? How to boost mitochondria in a safety way when there is disease or complex? Because based on my observation in the field, when the drug comes to the body, when the human has a disease, it's also curing this organ that destroys another organ. That's based on the observation from the people. So, could you please share the knowledge of how to boost the mitochondria in a safety way?
Oh, okay. So you are talking about boost the mitochondrial function.
Yeah.
As okay, that's a sort of a good idea. So number one, exercises. I think that's a proven way to make yourself health, right? Exercise certainly is the best way. Sometimes you can increase your oxygen intake, and which can certainly mitochondria is enriched with oxygen, so you can activate it. So the other is caloric restriction. So that's also the proven way to extend the lifespan, also health span. But people also propose the calorie restriction, also probably mitochondria involved. The other is certainly you have a way, either number one, can boost mitochondrial biogenesis, the growth, right? Like TFAM, anything can. Or you want to increase the mitochondrial quality control, to turn over, to degrade the damage to mitochondria. So that's probably the way to go. So easiest for you is approved exercise plus good diet.
Good diet.
Yeah, good. You see that all those metabolism goes through the mitochondria, right? You don't want to consume too much sugar, so which is certainly, you have the chance to leakage to generate a lot of free radical ROS. ROS certainly you have the way to oxidize different components, right, to potentially damage the mitochondria.
Thank you.
Yeah, thank you. What is that? Enzyme. Oh, okay. Everybody could have this bias among the respiratory chain, right? So I would say complex one is being the biggest one, right? It's oftentimes the reason I said complex one is not only my research concentration complex one, but also about 50% of chance those patients with respiratory chain disease have the defect complex 1. So that's also a reason. And also, interestingly, it looks like for the coding capacity. Right? So we have this, again, evolution speaking, you have all those mitochondria to begin with could be a bacteria. However, during this evolution, most of the genes, they shift to nuclear genome. They only retain 7, right? But 7 of them encoding for complex 1. So probably they also for a good reason to keep there. And so complex 1 actually is so big, 45 subunits. So there. Besides just for the electron transfer, we know in the yeast we have a single peptide called NDI1, which is enough for the electron transfer from NADH to ND1. So the single fact they keep 45 subunits, so must be doing something we don't know yet. So it's also, I think it's so not only for the potential importance, but also that means we probably need to pay more attention to explore this unknown territory to find more its role in keep us healthy, hopefully. Thank you.
Okay, are there any more questions? Apakah ada pertanyaan lagi dari teman-teman? Okay, while we wait for those who still have questions, can I ask a few questions?
Of course.
Okay, so I was very intrigued in the mitochondrial dysfunction. It amazed me how much of a small effect can actually lead to a very importance in the human body. You've talked about the dysfunction about the mitochondrial cells. I wanted to ask, is there any way to manipulate the mitochondrial dysfunction in hopes to help in the medical or maybe the science research? Are there any manipulation in the mitochondrial dysfunctions to help further researchers?
Yes.
Thank you so much.
So one thing, yes, thank you again for this excellent question. So to begin with, I missed the one thing is, as we know that electron transfer, right, if all the way to complex 1, 3, 4. So I also mentioned, so there is at complex 1, they have really high energy electron, right? So they go energy-wise, they go downhill, right? So during this process, high energy, relatively low energy, you have an energy conversion, which will be utilized upon 3-step remember potential. However, those electron transfer is not 100% proof that not every high energy electron stay within the electron transport chain. That means sometimes some of this energy probably will leave the electron transport chain early on at, say, complex 1 or complex 3. So at that time, because mitochondria is enriched oxygen, not at complex 4, they meet with oxygen, just generate water. If in early time they meet with oxygen, they will generate free radical or ROS, right? So people think ROS, they are hyperactive, they can oxidize DNA, probably mitochondrial DNA will end up with mutation, or oxidize protein, oxidize lipid, they all could have some adverse consequence. So that's one thing people do is they take antioxidant. For example, vitamin E, vitamin C, CoQ, right? CoQ we know is on top of the antioxidant or even melatonin. CoQ also is a substrate for respiratory chain. So those possible, one thing is antioxidant to neutralize free radical. Second is to boost mitochondrial electron transfer. So that's one way we can do it. Second is for those people already developed some mitochondrial dysfunctions, and now people trying to do mitochondrial transfer, which is put some new energetic mitochondrial. Then, of course, as I said, go exercises and eat healthy food. Thank you.
Okay, thank you so much, Professor, for your answer. Okay.
Can I have a question that I need the answer if you don't mind? Based on, please kindly share your knowledge, expertise. Based on your expertise and observation, is there any condition about permanent damage of mitochondria in human? And what is the causative permanent damage? Permanent damage. So its damage is permanent. The damage.
It's irreversible.
The condition of mitochondria is
Of irreversible damage.
irreversible. Yeah, it's different words. Yeah.
Yeah.
Sorry.
Yeah, of course, yes. So really, that's actually is an interesting idea, and that's also one of the theory for aging coming from, right? So we know there's multiple theory. So I guess everyone, of course, we biology study biomedicine included, even the lay people. Everybody should care about well-being, should care about the aging, right? Number one, why we will age eventually, and also there is a way to mitigate the aging or stay young as much as possible, right? So then also we came up with different theories to explain how aging happened, right? One theory, of course, is the DNA accumulation. The other is really by Denton Harmon, so who is chemistry by training. So aging really is an irreversible decline of function, right? You cannot do this and that. So you can think what could be the reason, right? So one thing is really the free radical. So free radical can oxidize the molecule so which is free radical can cause accumulation. So those damage could be irreversible, right? So then if you come up with this, really free radical is the can cause irreversible damage, which can lead into aging, then where does irreversible free radical generate, right? The majority ROS actually generate within mitochondria. Cause, again, just because the proximity, right, to cause mitochondrial delimitation or mitochondrial dysfunction respiratory chain. So really, and we did some research, and we measure, we isolate the mitochondria from different age, say muscle or brain from different age of the animal. Also, many other groups did it. We do find the decline of mitochondrial function during aging. So there is sort of like a well-accepted idea mitochondrial function really decline during aging because of this irreversible accumulated damage there.
Oke, karena teman-teman tidak ada pertanyaan, mungkin I will ask one last question to assure that everyone has already their answers. Is there any more questions? No? Okay. Ladies and gentlemen, as we come to the end of today's guest lecture, I would like to take a moment to express our deepest gratitude to Prof. Dr. Yidong Bai. Thank you for sharing your time, expertise, and inspiring body of work with us. Your contribution to the field of mitochondrial biology and medicine have not only broadened our scientific perspective, but also opened up new possibilities for future research and collaborations. To all of our participants, thank you for your enthusiasm, thoughtful questions, and active engagement throughout the sessions. Your presence made today's event meaningful and successful. We hope today's event has enriched your knowledge and inspired you in your academic and professional journey. Once again, thank you all for joining us. Stay curious, stay passionate, and see you at our next academic event. Have a wonderful day ahead.